Let's see these muscles actually practically in the macroscope. The first and ideal example is the tongue. The tongue contains numerous and endless skeletal muscles. In fact, it's one of the strongest muscles in the body. We're going to see two different conformations. We're going to see the cross sections and the longitudinal sections. Let's focus first on the cross sections. Typical picture is going to be this peripheral location of the nuclei of the muscle fibers. Of course, it's going to be in the periphery, and the center and the whole cytoplasm is going to be filled with thick and thin filaments. This is the longitudinal picture. We can see this, we can see the conformation in the longitudinal picture. If we take actually a proper focusing point and we tune properly with a fine adjustment knob, we can actually see these striations take place. Now, a very good eye can actually distinguish the Z discs and the bands specifically. But this, of course, is not necessary to happen. You don't have to do this. But if you actually pay close attention, you'll actually see them. Here we see this white area right here on the periphery, the two white areas. This is the eye band. And of course, in between the eye band, we can see the Z disc. This thicker, let's say, this anisotropic band, this denser morphology, this is the A band, the anisotropic band. And in between, you can find the H band, which is the small, let's say, loosened in the center of the A band. And inside here, you'll find the M line, not, of course, clearly visible through the macroscope. And again, this is the Z line, and all of this area is the I band. And again, and again, and again. These are the sarcomeres, and they repeat themselves endlessly from the one point at the beginning of the cell to the very, very ending of the cell. This picture actually will allow us to see, first off, completely and clearly the endomysium and the perimysium in this location. So here we can actually see the endomysium. This is the location where we can find the. Sorry, this is not the endomysium. This is the perimysium. The endomysium is in here in this location surrounding the muscle fiber. Now inside this very, very thin connective tissue, we can find reticular fibers, and sometimes we can actually be able to see the fibroblasts. Again, this is what the endomysium contains: reticular fibers and fibroblasts. Don't forget, reticular fibers is nothing more than a fancy word for collagen type 3. So, surrounding this, we're going to find the perimysium. This is going to be filled with, this is the thin connective tissue. And lastly, here, this is the epimysium, the top. On the top, this is the epimysium is continuous with a tendon, and we can see how this epimysium is actually invading. Look at follow the mouse. Epimysium, connective tissue, dense, and penetrates inside the structure of the fascicles. This is, of course, we've seen this picture before. This is the bone. This is specifically in this. If we zoom in here, we're going to find the ossification. And of course, this in the periphery, this is the dense connective tissue. If we zoom in, we can find the tendon, the continuation of the muscle with a tendon that will eventually lead to the connection of the tendon to the muscle, the myotendinous junction. So let's zoom in. In the top, again, we're going to find the epithelium. This is the parakeratinized epithelium. Always start from the epithelium. This is parakeratinized. Meaning what? What is parakeratinized? Meaning that this is here. Parakeratinized is a very specific type of epithelium, because we can actually see a thick part of the on the top. But so again, we can find the typical layers, the basale and so on and so on. And on the top, we find layers of dead cells. This is the keratin. This is the keratin layers. So this is the first information that we have. Then we zoom in and we go in the center, and this is where we find the cross section of the skeletal muscles. Again, the periphery located nuclei always, and the center is full of the center of the cell is going to be full with the thick and thin filaments. Now let's go to the longitudinal section. In fact, it's very easy to see, very easy to distinguish. This is the cross and this is the long. If we zoom in a lot, we're going to start to see the striations. This is actually why we call the striated muscle. So again, peripherally located nuclei. And again, we can, here for example, we can see a clear picture of striations. Very, very clear. These two dark lines, these are the Z discs. Unfortunately, I cannot zoom in more to show you details, but actually it's very, very easy to see the Z lines, the Z discs specifically. So Next up, we'll discuss about the cardiac muscle. The cardiac muscle is contains typically 1 to 2 nuclei. Of course, again, this is a product of the ones with the 2 nuclei are of course product of fusion of cells. But typically we're going to find one cell, one nucleus. And this nucleus is going to be typically in the center, centrally located. And we're going to see the different components right now inside. The cardiac muscle actually contains one structure that is very, very unique. It contains this intercalated disc. What does it mean? The intercalated disc is the junction point, the connection point between one cardiac muscle and the other cardiac muscle. Along this line, we're going to find a few very, very, very important structures. First, we have the desmosome. Desmosome is typically found, of course, in the epithelia, but of course here it's actually the function of the desmosome, both in epithelia and here in the intercalated disc, is to bring and to maintain tight cohesion, tight junction between one cell, cardiac cell, to the other cardiac cell. Along this connection point, this connecting point, we're going to find gap junctions. The gap junctions are inconceivably important when it comes to the function of the heart. Of course, the function of the heart is to beat rhythmically and to contract rhythmically with a specific tone and a specific strength. And most importantly, it has to contract at the same time a very, very large area of muscles in a specific sequence. But nonetheless, the atria and the ventricles should very, very rhythmically contract themselves fully. The whole atrium should be contracting at the same time, very, very organized. And for this to happen, we need to have the nerve impulse from the autonomic nervous system. The nerve impulse should be conducted and practically inducted in the whole length of the muscle at the same time. So, of course, the nerve impulse comes again through ions, through nerve depolarization, and these ions can actually pass through from one cardiac cell to the other cardiac cell through the gap junctions. So, the gap junctions are responsible for the rhythmic contraction and the organized contraction of the cardiac muscle cells. So, the differences here, of course, we have again transverse tubules. Transverse tubules again are the T tubules, we discussed them before. They're responsible for the simultaneous, let's say, release of calcium throughout the whole length of the muscle. Let's actually see them macroscopically. How does the heart look like? Here we have the cross section of the heart, and here are the longitudinal section of the heart. The cross section. First, we notice a difference. The centrically located nucleus, not peripherally, as the skeletal muscle. And the second we have is this is the longitudinal section. Again, the nucleus is centrally placed. So let's zoom in and try to. So here we can find the cross section first. And how do we know there's a cross section? Because we can see the centrally placed nucleus and the thick, let's say, circular shape of the whole cardiac muscle. So in this point, we cannot get much information from the structure. So ideally, we can see this intercalated disc and the other structures in the longitudinal section. So let's search for one. Let's look for one. First of all, let's actually discuss what are these. These are the adipocytes. This is the white blood. This is the white adipose tissue. This is again a common finding in the human heart. The more, of course, this is why we actually have to very, very be careful of the fat content in the human body, because the more fat we have, that there is a high amount of deposition inside the heart and actually produces and exerts extra additional strain to our heart. So this is the longitudinal section of the heart, and let's zoom in. Lovely. These lines, this is, for example, the perfect example of the intercalated disc. Same here. And again, inside this intercalated disc, we can easily see one common feature: the two different components, the desmosomes and the gap junctions. This surrounding the nucleus of long-living cells, there is one common thing. Long-living cells are two different long-living cells in your human body, most commonly, let's say. The neurons, and the cardiac muscle cells. These cells do not regenerate, generally speaking, and do not keep on mitotically dividing throughout life. So these cells are long-living cells and they last us for life, the whole lifetime. These cells, we discussed that in the beginning in the cytology again, about the tertiary lysosomes. The tertiary lysosomes, again, are the lysosomes, the portions of the fused phagosomes with the lysosomes that cannot be digested. Again, This is the end result in a long-living cell. When you don't have the potential to practically regenerate, it's very common to have these lipofucsin pigments. These tiny holes right here with this brown yellowish hue are actually the lipofucsin pigments. And this is common findings. This is one of the common findings in the long-living cells. I think I have a picture here that actually shows it a bit better. There we go. Again, this is this brown yellow structure. This is the lipofucsin pigment. Again, this is tertiary lysosomes. The heart again contains the same filaments, thick actin and myosin filaments. The contraction logic is the same. Again, we have the transverse tubules. Again, the structure of contraction and the skeletal fibers are the muscle fibers are very similar. In fact, almost identical. So let's talk about the smooth muscle cells. Now, the smooth muscle cells have something unique, something different. They do not work in the same way that the other muscles do. Again, of course, we can find both of these, on all three muscles, we can find the thick and thin filaments. That's true. But here, there is a different modulating and regulating machinery. Here we have that these actin filaments do not contain troponin. This is because the constriction of the smooth muscle is of a different logic. The construction of the smooth muscle fibers is not continuous. It's not intermittent. It's not controlled by our human mind, our human minds, and they're always in some form of either contracted or non-contracted, somewhere in between. There is not this logic of all or none. In the skeletal muscle, in the cardiac muscle, there's all or none. Either there's contraction or there's not. Here we find something in between, meaning that these cells are always somehow contracted or somehow relaxed, but not fully contracted or fully relaxed. This is actually the mode of many smooth muscle fibers, and this is actually how most of our GIT system works. The gastrointestinal tube, that's how it works with this type of movement. So these units lack troponin, so we don't have this very strict, let's say, controlling point. They contain this calmodulin and this myosin light chain kinase. Calmodulin is nothing more than just the molecule that again practically receives calcium, and same as the myosin light chain kinase. These molecules somehow regulate the contraction of this muscle, the smooth muscles. And of course, there are no striations because there is no presence of the sarcomeres here. Let's talk a bit about how this actually contraction takes place. Well, the smooth muscle cells contain dense bodies. The dense bodies are points of attachment for the thick and thin filaments. Now, these thin and thick filaments are connected between the one cell and the other cell. So the dense bodies actually have dual function, both to control and to attach on them the thick and thin filaments, but also to connect more than one, to connect the different numbers of smooth muscle cells. And of course, we need to understand that the constriction of the smooth muscle cells, let's say for example in the GIT, in the intestine, is in a layer. We'll see how it actually looks like, but the logic is that a whole layer has to contract somehow, not fully rhythmically, but somehow has to connect with one another. And the way that this is done is because of the dense bodies. So this is an actual picture of how these actin and myosin filaments are again connected. Of course, they have similar connecting points. The actin is the same, tropomyosin is the same, but there is no troponin unit. So the tropomyosin is always somehow connected to the actin and is released through the function of calmodulin and the myosin light chain kinase. So these points, this is the dense body connecting both the actin, the thin and thick filaments, and connects two different cells. This is the one cell, this is the second cell, and allows for at the same time this mild contraction. A key characteristic of the contraction of the smooth muscle fiber is the conformational change of the whole cell, also the nucleus as well. This is how we can identify easily, and we can easily see that this cell is a smooth muscle cell, not a fibroblast, because the nucleus in the fibrocytes, or the fibrocyte of the smooth and the smooth muscle cell and the fibrocyte are similar. And the way to easily differentiate the layers and the different cells is when the contraction takes place, the shape of the nucleus takes an S-shaped conformation, let's call it. Let's see it actually macroscopically. So again, this is the intestinum tenue. This is the small intestine. If we zoom in again, this is the tunica. This is not the tunica. Sorry, this is the mucosa, and beneath it we're gonna find the muscle layers. So this is the cross section of the smooth muscles. Again, we can see the centrally located nucleus, and we can see many of them together, like practically connected with one another. And this is the longitudinal section of the smooth muscles. So this shape of the nucleus is actually called cigar shape. You can actually see this term in tests or in some books. It's a very common characteristic because it actually looks like a cigar. Unfortunately, I do not see an S-shaped nucleus right here. Again, this is the mucosa, submucosa, and now let's go to the muscle layer, muscularis. So again, all of this layer is typically, this is the muscular layer. So let's try to find a nucleus that is contracted. Here there is some sort of contraction, mild, not major. Again, we can see this, let's say this movement, this change in the shape right here. There we go. Look at the shape, look at the change, look at the density of the nucleus right here. Way different than the typical cigar shape. So again, this is the difference between the smooth muscles. The smooth muscle actually has this not only all or none, it has this intermediate contraction level and allows for the whole layer to somehow fully contract at the same time. So imagine all of this layer is connected somehow in a specific way through the dense bodies, and they can contract mildly in a very, very rhythmic way and allow for the food to pass through. So that's it, guys. That's it for today.